So I welcome you all to another important lecture. And in today's class, we will look on toward the structure of the bacteria, and as far as these bacteria are to be concerned, so they do not have a eukaryotic cells, but rather than they have the prokaryotic cells with us. As far as the classification system is concerned, so we have got major of three domains. We have the eukarya, then we have got the prokaryotes, and then we have got the archaea. So these bacteria that we have with us, so they belong to their unique domain, and that domain is prokaryotes. And as far as the prokaryotes are concerned, so for any of the bacteria that we have got in the environment, so some structures they would be existing or they would be available in all the typical bacteria, but there would be coming up of some other cellular structures that would be present in some bacteria and not in all the bacteria that we have. Now, if I were to tell you, so for any typical bacteria, so there would be the presence of cell wall, then they would have their cell membrane. They do not have their genetic material enclosed by the nucleus, but rather than they have in themselves a kind of a DNA, and that DNA is a circular DNA. And the region where this circular DNA would be lying, so we refer to this as nucleoid region, and this is again not enclosed by the nucleus, but rather than we say that this lies naked within the cytoplasm of the bacteria. So we have the cytoplasm as well, and for the bacteria strictly, their ribosomes they would be 70s ribosomes. For the eukaryotic cells, the ribosomes were 70s and 80s. The 70s were available within the mitochondria and the chloroplast, and the 80s ribosomes they were available on the membrane of the rough endoplasmic reticulum, and any of the ribosomes that we would have in the cytoplasm, the ribosomes were 80s. But for the bacteria strictly, their ribosomes would be the smaller 70s ribosomes. Now, there happens to be some other bacteria, and these bacteria they are unique with respect to this, that along with these typical structures that we see in the other bacteria, so they have some enfoldings for their cell surface membranes. And not only that they have their circular DNA, but they would also be having these plasmids, and these plasmids they are termed as being serving as an extra genetic material. And we will see in a while that what these plasmids they actually do for the bacteria. Some of the bacteria they possess these pili as well, and they would be responsible for the type of the sexual reproduction that we see in the bacteria, and that is the conjugation. And then in fewer bacteria or in some of the motile bacteria that have an ability to move, so they have got these flagellum in them. Okay, so this is something that you need to remember in terms of some small amount or fewer, you could say, bacteria that would have these extra components in them. Now, I would quickly take you to be understanding the structure of this bacteria that I have drawn for you. So here you see that I have got their cell wall, then they would have their cell membrane, and then there would be the presence of the circular DNA, and the place where the circular DNA would be lying, this is termed as nucleoid. Moving onward, they have got here would be their cytoplasm, and they have in themselves these ribosomes. Now, these are the characteristics that would be available in all the bacteria. Now, for the some bacteria, there would be the lying of something outside of their cell wall, and this could be the slime layer, it could also be in the form of capsule. And then they have got the enfoldings of their cell surface membrane. And some of the bacteria, they have got these extensions out of them, and these would be the flagellum, we say. And then along with the circular DNA, there would be the presence of this extra genetic material. And this extra genetic material is what we entitle them as the plasmids. And then there would be the presence of these structures, and these structures they are entitled as pili. Now, let us just try to see each and every component that what this is doing for the bacteria. So here you see that we have got their cell wall, and unlike the plant cell, which was made up of the cellulose, the bacterial cell wall would be made up of peptidoglycan. That would be a complex of the protein and the sugars. Now, moving downward, their cell membrane would again be acting as a selectively permeable or partially permeable membrane, and it would be regulating that which substances would be allowed to enter into the bacterial cell and out of the bacterial cell. For their cytoplasm, the unique thing is that it does not have any of the double membrane organelles. By the double membrane organelles, we mean the nucleus, the mitochondria, and the chloroplast. So the bacteria, they lack the double membrane organelle in their cytoplasm, and this cytoplasm would be acting as a place where most of the bacterial chemical reactions would be happening. Now, since the bacteria they are living, so they would also be requiring the ribosomes because they have to be making the protein. So from their circular DNA, the transcription of the gene would happen. They would likewise make the messenger RNA, and that would be carried to the ribosomes. And when it would come to the ribosomes, so these bacteria they would make the proteins that are important for the bacterial survival, like the enzymes that the bacteria would require, or some of the membrane proteins that the bacteria would be requiring. So they would be made by their smaller ribosomes, and they are the 70th ribosomes. So this is the detail of the typical structures that we have in all of the bacteria. Now, if I were to talk about the unique features that we see in some of the bacteria, so on the outside of the cell wall, now here you see that we have got this layer, and this could be capsule and this could be the slime layer. Now let us see that how come the capsule and the slime layer they are different from. As far as the capsule is to be bothered about, so this would be forming a definite structure outside of the cell wall in some of the bacteria, and this is by composition made up of the complex sugars and their polysaccharides. And how this capsule is different from the slime layer, that it is not easily washed away. But if on the outside of the bacterial cell wall, if we have got this slime layer, so this has got more diffuse structure, that means as that they can easily washed away. No matter if we have the capsule or if we have the slime layer for the bacteria, so the role which they would be performing is that they would be saving or protecting the bacteria from drying off, because if the bacteria would dry off, for sure they would be killed. Now, further moving on, now here you see that for some of the bacteria, we have got these enfoldings, or these, you could say, extensions that get out of their cells. So they are referred to as a flagellum. And this flagellum does not has a complex structure as we have got in the cells of the eukaryotes. This has a simple structure. And the flagellum role would be that it would allow the bacteria to be moving, or for the motile bacteria, these flagellums would be available. Now, in terms of the enfoldings of the cell membranes that we have got, so these enfoldings of the cell membrane, now they could be available in some photosynthetic, you could say, kind of the bacteria, those who can make their own food. And likewise, there is another category of the bacteria which are nitrogen fixing bacteria. And what the nitrogen fixing bacteria they do is that they would convert the atmospheric nitrogen gas into the nitrate ions. And this conversion of the nitrogen gas into the nitrate ions, now this would require these cell membranes enfoldings, and that would be unique to these nitrogen fixing bacteria. Now, moving onward, in some of the bacteria, along with their primary DNA molecule which they have in them, which is circular, so some of the bacteria they have got this unique structure in them, and this is referred to as a plasmid. And as far as a plasmid is to be bothered about or to be discussed, so we say this is extra genetic material, because the primary genetic material of the bacteria is their circular DNA. Now the situation is that what this plasmid actually do for the bacteria. So the plasmid they have got the genes for the antibiotic resistance. You see that at any point when we people get ill, so we are eating or we are ingesting or we are taking the drugs that are antibiotics, and those antibiotics they have an ability to kill these bacteria. But bacteria, being the living organisms, they develop a kind of a situation, and this is termed as an antibiotic resistance phenomenon, where the plasmid would carry the genetic information to make some enzymes or to make some transport proteins, and that would degrade the antibiotic, and in that way, the antibiotic would not be killed. The other situation is that these plasmids they can also transfer or can be shared between the two bacteria, and for that there would be the need of these structures, and these structures they are the pili with us. Okay, the pili they have the role not only allowing the bacteria to attach to their host, but at the same time they can also act as a mode of transmission of the plasmids from one bacteria to another bacteria. Now, this pili has got a role in determining the sexual reproduction for the bacteria, which we term this as the conjugation. Now you see, you know it well, that the reproduction in the bacteria that we have got, so they could be further split into the sexual or the asexual reproduction. So now you see that here we had one bacteria, and originally this do not have the plasmid in it. And now this bacteria has got this extra genetic material, which is a plasmid with us. And if the plasmid has to transfer from one bacteria to another bacteria, so the pili are being utilized for that purpose. So this is conjugation, and this is a type of the sexual reproduction that we have got for the bacteria. So I hope you all would be clear now with understanding the structure of this bacteria or the prokaryotic cell.